Okay, this is Mr. Block, and I'm going to be gone today for biology. So you're to start off class, you're going to do a lab, and usually I don't like to be gone when we're doing labs, but I know you guys can handle it. So we're doing a yeast lab where you're going to be learning. We're going to be learning about populations and how they change over time. And so we're going to be looking at a population of yeast. Yeast grow through binary fission or reproduce through binary fission. So about every 20 minutes, they can divide and make another copy of themselves. So you can start out with one, and 20 minutes later, you'll have two, and 40 minutes, you'll have four, and 8, and 16, and so on and so forth. So we're going to see how their population changes over time and what kind of growth we see. All right, so the lab today, you're going to be basically setting them up in their environment, and then you are going to be making a first day count. Okay, so our yeast will be in a beaker like this. It'll look kind of yellow. Whoever's the first one that gets there for your class period, make sure you stir it up a little bit so that we get a good mixture of yeast and water. Okay, what we're going to put them in to grow is grape juice. So they like the sugar. They eat the sugar and they produce carbon dioxide, and that is how yeast make bread rise. Okay, so what you're going to do, the instructions for this lab are in Live Binders, chapter 4, and it'll say yeast lab directions. Okay, so I'm going to go through what you're going to do, but you need to look at the instructions as well. So it says to take 19 milliliters of the grape juice, and you're going to measure that in a graduated cylinder, and then you're going to put it in a plastic cup. Okay, the plastic cups will be on one of the lab stations. Okay, you'll pour that in there. I'm not going to measure it right now because I don't want to spill grape juice all over my desk. So you'll put 19 milliliters in the cup. Then it says to take 25 drops of the yeast solution. Okay, 25 drops equals 1 milliliter. So, we have our grape juice. I didn't measure that, but you get the idea. Then you'll take some with your eyedropper, and you're going to put 25 drops into the grape juice. Okay, so that's going to make a grape juice yeast solution. We use 25 drops because that equals 1 milliliters plus 19 milliliters of grape juice gives you 20 total. You're going to take a toothpick. You're going to stir that up really, really well to make sure we get a good mixture. Okay, we're then going to put 1 drop of the yeast grape juice solution. Okay, if you're using the same eyedropper, you want to rinse it out with water really, really well. Otherwise, you'll have a bunch of yeast in there, and there's going to be too many to count. So make sure you use water and suck up water and all that stuff in the sink. Okay, you'll take 1 drop of your solution. You'll put it on your slide like this, and then you'll take your cover slip and you'll put that over the top. Okay. Now, then you're going to put it under the microscope and start under medium power, the yellow, and then eventually you'll need to get to the high power, the longest lens, to measure them. Otherwise, they're going to be too small to count. Okay, and it says in your instructions to make 5 different counts. Okay, so what does that mean? Basically, here is your slide. Here's your cover slip. The first time you're going to draw, or you're going to put your microscope right in the middle and count whatever you get. All right. Then you'll move your slide a little bit, and then you'll count like in a corner area, and then another corner, and then another corner, and another corner. So basically, you're going to count 5 different spots on the cover slip, and those will be your 5 areas. There's a data table in Live Binders that you will use. It says yeast lab data table for your group. You'll save a copy and share it, put all your data into that. Okay. When you are all done, you'll put some aluminum foil over the top of your solution. You'll put it in the fume hood. You'll see a bunch of other ones there. And then we'll let that grow over the next 3 or 4 days. And then we'll count them again and see how their population changed. Make sure you put your name on the aluminum foil so we know which is yours. And then when you're totally done, clean up your lab station, rinse out your beakers and your graduated cylinders, put the slides on a clean paper towel, and that should be it for the lab. All right, after the lab, you're going to watch a video presentation of section 4.1, and I will put that video, I will send you the link to that video as well.